Today there are many things which we don't know about brains.
This project will give us an unprecedented knowledge on how the brain operates and how it is affected by brain disorders. Alzheimer's disease is an example of neurodegenerative disorders, and for which we don't have markers that would enable us to diagnose at an early stage. And diagnosis is made mainly based on symptoms that are not very specific, and the confirmation, unfortunately, has to be done post-mortem.
We use the conventional light microscopy, with which we can see on cellular level changes in the brain. But when we want to see more details, we can change our methods and use light sheet microscopy.
The issue with traditional techniques is that they are done on small samples and we miss out the large connected brain circuits. This project aims at addressing this challenge and at imaging brain circuits at the cellular level in the whole organ.
Some of the challenges we face today with imaging larger samples are first on the microscopy side. The previous generation of light sheet microscopes were illuminating the samples from the side. But today the next generation of light sheet microscopes are illuminating from above. So now we can fit larger samples like this book, and we're illuminating them from above, from both sides. This new generation of light sheet microscopes allows us to image a whole human brain in only 10 to 12 sections.
We are using a series of techniques in order to make the tissue clear and be able to study what's inside the tissue. We are doing this by removing the lipids present in the tissue. Once the image has been done, what we can have is an entire image brain with the different cell types. And of course, we can start atlasing, so really studying the different cell types in different brain regions.
An important part of the project is to be able to compare several brains, for example, a control one and a diseased brain. So to do that, we need to align both brains on the same universal map that will highlight the different brain regions with different colors. And then we can, for example, count the different cells inside each region and compare between both brains to understand what was happening at the cellular level. So in order to analyze this data, we use machine learning techniques and deep learning techniques to extract the information that we need, for example, the cell position in the brains. So in order to image a whole human brain with a very high resolution, we would need to generate as much data as the entire YouTube storage. In order to accelerate the storage and the processing, we need to find a smart compression algorithms that will compress by 100 to 1,000 times while still allowing to process this data faster.
We believe this project will allow us to find precursors for important brain disorders, and from there we will be able to find ways to revert them.